Hello, my name is Jiri Aubrecht, and I'm leading a biomarker group at Sarepta and also function as a PSTC industry co-director. So I'll be talking today in our webinar about a glutamate dehydrogenase and describe you a journey of a biomarker from qualification to potential even improve a medical care and medical practice. On the next slide, on the next slide, I'll touch on why we actually started this. There's an unmet medical need where in drug development to monitor liver safety when we are developing therapies for patients in neuromuscular diseases, there's a need for a liver-specific biomarkers of liver injury. And the same thing also is for a clinical practice where a diagnosis of liver impairment in subject with muscle diseases is difficult due to a lack of specificity of biomarkers which we are using, which is ALT. To demonstrate that, on next slide, on the left panel, you see that the ALT closely correlate with a marker of muscle injury or muscle damage, which is creatine kinase. And therefore, changes in the muscle disease or muscle wasting go to release of creatine kinase and ALT, and then the changes in the ALT can potentially mask the effect of treatment or disease in those subjects with underlying muscle disease on liver, and then can be diagnosed, which causes problem in drug development and in clinical practice. So also some people say that you can use some alternative biomarkers like glutamate, like GGT, gamma glutamate transferase. But gamma glutamate transferase, as you can see on the right panel, is a specific to a bile duct and does not reflect a hepatocellular injury as the transaminases. So therefore, a development of a liver-specific or hepatocyte-specific marker is important. So we ask ourselves early in the project whether glutamate dehydrogenase can actually rectify the shortcoming of ALT. Glutamine dehydrogenase, GLDH, is very well known, a protein, and its role in metabolism. And its tissue distribution is also known, where most of the glutamate dehydrogenase is actually in the liver. There's a small amount in kidney and pancreas and intestinal mucosa, but only traces of GLDH are present in muscle. So therefore, it seemed to us as a good biomarker candidate to evaluate liver injury in subjects with underlying muscle disease. In addition to that, there's an assay available, which is quite relatively easy and high throughput to perform. And there are some data where showing GLDH as a liver-specific marker or a marker for liver injury in preclinical species. In order to use GLDH in clinic, there are gaps, and those gaps are what are the ranges? What is the distribution of values and influence of genders, age, or ethnicities? There's a lack of systematic evaluation of GLDH as a biomarker of liver injury in humans, period. We don't know about interpretation of the data, cutoffs of concern, and therefore there's was really a limited acceptance or no acceptance of GLDH by regulatory agencies in drug development for monitoring of liver injury. So we set out. This was the unmet need, and we set out to define a context of use and qualify a GLDH. And as a context of use, we really pin it to as a marker which is suitable for evaluating liver injury in patients that have where ALT results are confounded due to a variety of reasons, such as muscle disease. And in that cases, the GLDH would be preferable and used in lieu of ALT with in context of other biomarkers like bilirubin and so for detection and evaluation of liver injury. So now, how did we approach the qualification process? And we chose a translational approach, a series of non-clinical and clinical studies. The advantage of the non-clinical studies allows us to anchor the performance of the GLDH2 histology, because we can get, in these studies, we can get tissue samples and evaluate the actual histopathology, the effect of compounds or disease in animals. This, of course, it's very difficult to do or even impossible to do in clinic. And therefore, we establish using diseases as an approximate of chemically induced muscle injury. And the clinical studies are allowed us to confirm a non-clinical biomarker, the non-clinical biomarker performance, and establish all the caveats and all these levels, cutoff levels for interpretation in humans. So just a couple of words about the analysis or the studies which we had in clinic, that where we leveraged samples, leftover samples from healthy subjects in clinical trials, and leftover samples from patients in clinic, and which we received or collaborated with the University of Michigan. And that allow us to work closely with the physicians and determine in clinic the extent of the liver injury by medical adjudication. And we also utilized a biochemical criterion for liver injury, which is well accepted. And so we call that approach a prospective analysis of banked real-life samples and approximation of the disease-induced injury to a chemical-induced injury. Was the way how we approach the qualification. So we had three major objectives for the clinical qualification of GLDH. First, we decided to establish a reference range for healthy subjects and evaluate the influence of sexes and age groups. The second was make sure or develop data sets that we can establish GLDH as a sensitive biomarker of liver injury, at least as sensitive as ALT. And again, done that a series of non-clinical studies where we were able to use a variety of hepatotoxicants and anchor the performance of GLDH to histopathology. And in clinical studies, we used the diseases, the samples from subjects with a variety of liver impairments to evaluate the concordance of GLDH and ALT, and then we also had several patients with accidental acetaminophen overdose. And then the third objective was to understand the caveats. So first objective is to get the reference ranges and the variability across the population. The second objective was, do we have a sensitive detection of liver injury? And third objective is the caveats. So determine what is the specificity of the liver injury, what we need to take care of, or what we need to keep in mind when we interpreting the data. So that means evaluate the ability of GLDH to differentiate muscle or liver injury in non-clinical species, and in clinic, and evaluate liver specificity in subject with of GLDH in subject with Duchenne dystrophy, myositis, rhabdomyolysis, and other muscle impairments, and also try to determine whether GLDH can detect onset of liver injury in rhabdomyolysis patients, because we were collecting different cases where if we were lucky, we could have actually see a liver injury on the background of muscle injury in humans. So to the objective number one, our study included, which was published almost now 8 years ago, which included almost 400 subjects across a variety of age groups, and both in males and females. We were able to establish that the GLDH is not affected by the sexes, and not affected even by the ages. So it's really a big advantage of GLDH being very robust, and the reference range, which we confirm in subsequent studies, really is up to 10 units per liter. So now, evaluating the performance of GLDH in non-clinical studies, we were able to do 32 or examine effect of 32 model agents, a variety of toxicants, and we're able to assess the performance by using a ROC curves, which is on the left side, where we compare sensitivity with specificity, and the area under the curve is a measure of a performance of a biomarker being a sensitive and specific. So the higher the area under the curve, it's the better performance, better specificity and sensitivity. And on these studies, GLDH actually outperform ALT. I would call it that GLDH definitely is not inferior to ALT in non-clinical studies. So in clinic, in our paper from 2013, we surveyed more than 700 subjects, and a healthy subject and the subject with variety of liver impairments, which have increases of ALT in the wide range increases of ALT, and also several cases of acetaminophen poisoning. And as you can see, we were interested into a correlation between GLDH and ALT, and there is a nice linear correlation between those levels. And we used a biochemical criterion for definition of liver injury. Obviously, we couldn't use histopathology, because that's not available, and constructed the same ROC curves. And as you can see on the panel below, on the lower panel, the ROC, the area under the curves curve for GLDH is 0.98, and the ideal ROC is 1. So it's probably as good as it gets. So that's kind of gave us a confidence that GLDH is sensitive and specific biomarker of liver injury, at least, if not better, than alanine aminotransferase. So now we were looking at whether GLDH is not affected by a muscle injury, and we look at it in a variety of ways. We'll look at it that we compare GLDH levels in the subject with Duchenne muscular dystrophy with healthy subjects, and that's the panel A. And then on the panel B, our data from subjects with variety of acute muscle impairments, myositis, rhabdomyolysis, and so on. On the upper panel, where we studied the subject with Duchenne muscular dystrophy and compared with the healthies, you can see that these subjects with DMD has about 20-fold increase of ALT, and that ALT is coming out of muscle. That increase, and that it all fits together with the levels of creatine kinase, which is highly increased in those subjects, documenting that they definitely have a muscle injury or muscle impairment wasting. All healthy subjects, the healthy boys, which are matched, which were in the same age group as the DMD boys, and the healthy subjects had a normal level of ALT and also a normal level of CK. So if you look at the GLDH, you can see that the GLDH levels in DMD boys was indistinguishable from the healthy boys or healthy subjects. Just to document what levels you actually see with GLDH in subject with acetaminophen poisoning, that's the lowest row in the panel A, and you can see the difference and appreciate the difference. So now in the panel B, that describes about 150 subjects and healthies that have a variety of acute muscle impairments like myositis or rhabdomyolysis. And on the lower left panel, you can clearly see that the CK and ALT correlates, and it's what it should, because the ALT is coming from the muscle as well as the CK. And if you look at the lower right panel, the correlation between CK and GLDH is not there. So that means that GLDH is not affected by the muscle injury in subjects with acute muscle injuries also. So now, as I told you, we were looking and collecting cases of muscle injuries and hospitalized patients, and measured GLDH. And in one case, we were actually lucky because there was a case of a subject who was admitted to the hospital and developed rhabdomyolysis, which is documented by the rapid increase of CK, ALT, and AST. And as you can see, the GLDH was within normal levels. I had to make sure that everybody knows that it's a fault of change is the y-axis, and it's logarithmic scale. So up to a day about 22. And day 22, the subject suffered hypoxic crisis, need to be incubated, and later recovered. But it led to a ischemic damage of liver, which is nicely reflected with the increase of GLDH, which is the green line. And just, got to say that this is a only way how you can detect it. All these cases are undetected without a liver-specific biomarker of hepatocellular injury. So that's the advantage of GLDH in clinical practice too. So, of course, we wanted to look at the performance of GLDH on the background of muscle injury, and do a more systematic study and compare with the histology, which we couldn't do in those cases of rhabdomyolysis, which I presented. That case of rhabdomyolysis I presented on the previous slide. So we helped ourselves with a animal model of muscle disease, and we used the Duchenne dystrophy mice as an animal model, and we treated them with a toxic dose of acetaminophen, and we evaluated the histopathology. And as you can see, that the control, which are the green points, have no effect on histology. The histology is negative, which is on the histology results are depicted on the x-axis. And as you can see also that there are 2 treated animals which didn't have any detectable histological findings after treatment with acetaminophen, and that's normal. Sometimes acetaminophen doesn't work at this particular dose, doesn't cause 100% damage in all animals. That's normal. And then you can see that animals with a minimal damage, histologically detectable, had about the same levels of ALT as the ones they didn't. Only a marked lesions, induction of marked lesions led to an increase, appreciable increase of ALT, and that's at the moment that the liver are damaged so much that they overwhelm the muscle release of the ALT. But on the other hand, the lower panel is the GLDH, and here you can see that again those 2 mice which were treated with APAP and didn't have any histopathology, those were within normal limits with the GLDH, but even the animals with minimal injury detectable histopathologically had a significant increase of GLDH, as well as the mice that were affected the most. So then we had an opportunity to actually get a handle on samples from a large population trial where they were evaluating patients with cardiovascular risk factors. It was 3,400 or 3,600 patients, and we could measure all GLDH level in those patients and ask questions. For example, correlate the GLDH and ALT levels with alcohol consumption, which is the panel, which is the upper panel. And as you can see, the increase of alcohol consumption, which is self-reported, which might be probably bigger than it's here. You can clearly see that about 21 units and above of alcohol leads to an increase of levels of ALT, and the same you can actually happens with the GLDH, which is on the right side. And what was also interesting, that subjects treated with statins in general had a statistically significant increase of ALT, small but statistically significant increase ALT. All these increases are not above an upper limit normal or anything like that, but within normal levels. But as a group, they are differentiated from the ones they don't get, didn't take statins. And in the case of GLDH, we see just a trend of an increase in statin users. They're not, but that's not statistically significant. So we of course cannot exclude that the effect of statins on people is also on the liver and was described, so that which might cause that little non-statistically significant increase. But most likely that increase in ALT is the effect on muscles. So furthermore, we were able to, in that particular trial, we were able to look at association of levels or increases with variety of factors like diabetes, and that was very eye-opening. For example, it confirmed already known association that patients with diabetes have a higher level ALT than patients than healthy patients. And this table, I use p-values to document the presence or absence of any of these. We also saw what has been shown before, that females usually have a little higher ALT levels, and then also the ALT levels increases with age. And what was striking, that a body mass index is associated with a higher level of ALT and so on, and even smoking subjects have a higher level of ALT. But then looking at GLDH, the GLDH it's really unchanged in diabetes. It's really unchanged with body mass index, and in age and genders, so don't play a role. And that provides a really food for thought how to use GLDH. That GLDH might be really a good a marker for liver health, especially useful in areas like non-alcoholic, fatty liver fatty disease, and so on, and diabetes, to really look at and evaluate the liver health. So now that kind of provides a segue to the clinic and how we can use it. So regarding the status of the qualification as a drug development tool, we are GLDH has been used quite a bit in clinical trials, supporting specific clinical trials for new modalities for treatment of muscle diseases. I just looked today at the clinicaltrials.gov and found and searched with GLDH and found 13 studies in that database. Most of them are are for treatment of Duchenne dystrophy, and notwithstanding a even larger category are their studies. They study a non-alcoholic fatty liver disease and dyslipidemia and liver disease in general. So, which is kind of interesting that we started with the muscle diseases, and it seems like by analyzing the clinicaltrials.gov, people noticed and start using the biomarker also as that biomarker of liver health. And we pursue formal qualification with the FDA and EMA, and we are really in the final stages submitting the final qualification package and expecting having a qualification decision in 2022. So just putting all thing together, that journey that we started with a drug development or using GLDH in drug development of modalities for treating neuromuscular diseases, which is the panel 2, and using it for monitoring of liver safety, which provide the advantages which ALT does not have, to a diagnosis of disease in patients with muscle diseases, as we shown in our with the case of the patient with rhabdomyolysis, and also as a biomarker of liver health for chronic. Could be applicable for chronic liver diseases, and based on the analysis of the clinicaltrials.gov, it seems that's what's going on. So now, what is limiting actually the use of GLDH in clinic? If you think about and having a patient in mind, so why people don't use GLDH more in clinical setting from that drug development setting into clinic. And it's really the biggest obstacle is, of course, we need to have more data, and as the data are coming on, people get more interested, the physicians and the patients. But the wide use of GLDH in clinic is limited by we don't have available a proper GLDH in vitro diagnostics or diagnostics which has a designation, in vitro diagnostic designation in the United States. And that requires the 510K submission process approvals for devices, which at this point we are in a unclear market size and justifying the investments to the diagnostic companies to actually move forward with that. And so therefore, we ask ourselves, can we actually help as a consortium, and can we help with our qualification data sets to support the 510K filing, and get a collaboration with the diagnostic partner is essential. And when we've been working on it for some time now, that we can bring GLDH in collaboration with actually Roche Diagnostics to patients. And how we do that? Leveraging really a partnership between the diagnostic company, and we are very grateful for the collaboration with Roche Diagnostics, with the PSTC consortium, the biomarker consortium, and the Duchenne de-risk consortium, and bringing us all together with the goal that we would be able to get GLDH as an in vitro diagnostic to every laboratory, every clinical laboratory, around the world. And we're putting all our unique abilities and expertise in play, and working together to get that. It's been a very interesting and eye-opening project for us, and hopefully we'll get there. So I would like to end my presentation with acknowledging my collaborators. Most of the work has been done by collaborators from Pfizer, University of Michigan Ken Johnson, and Russell were absolutely essential in the bank samples, real-life bank samples approach, and of course all the partners at PSTC and John Michael Sarah and Nick King playing a major role in this process, to shepherding it and providing the leadership to get us to the qualification. Thank you very much for your attention, and hopefully I see you at the panel session.